Hi, welcome once again to our YouTube channel. Thanks for subscribing to our channel. We really appreciate you. It's nice that you are coming along with us all along. We really appreciate that. For those that are yet to subscribe, please don't fail to do so. Kindly click on the subscription button and also click on the notification button so you get easily notified of subsequent uploads when we upload them. Also, try as much as possible to like our content, share them, and please don't forget to send in your comments. We would love to hear from you. Today what we actually want to talk about is dissection. We actually want to carry out dissection in biology, dissection of a particular experimental animal. And this is the experimental animal that we will be using. We will be using a rabbit. We will be using a rabbit. The scientific name is Oryctolagus cuniculus. That is the scientific name, the Oryctolagus cuniculus. That is the scientific name of a rabbit. So it is a rabbit that we're going to be using for our experimental proceedings today. So I'll quickly encourage you to try as much as possible to follow us with everything that we will be doing so you get to understand the series of steps that will be required in carrying out what dissection is. Thank you very much. Alright, in carrying out this particular experiment, what we'll be doing first and foremost, we need to place some papers on our board. So let me quickly do that. I'll be placing some papers on my board right now. So I want to use these papers for my experiment. So let me just quickly pin my papers to my board. We don't want to smear our board with the blood of the animal. That is why we need to use a white plain paper. So that is what we are doing right now. Alright, so we have placed our paper on our board. Now, we need to actually sedate the animal. We don't just expose our animals to knife. We don't just expose them to knife, so we need to sedate them. So that is what we are going to be doing right now. And in the process of sedating our animal, I will be using a particular substance called chloroform. As you can see here, it is an anesthetic. So we are going to use chloroform to anesthesize our animal to make it unconscious for us to actually carry out our experiments. So that is what I am doing now. So I'm actually going to use this chloroform to sedate our animal. So I am using this as an example of what we call a crucible. You're actually supposed to use a crucible to sedate our animal, exposing the animal to the chloroform, which is going to in turn anesthesize the animal, causing it to become unconscious. So that is what we are doing right now. In a couple of minutes, you will discover that the animal will sleep off when it has inhaled the gas of the chloroform. The animal is still conscious right now, but soon it will sleep off. So here we have some set of the dissecting instruments. So, but I'll be using these ones that I've placed here, majorly for what we have to do today. Alright, so the animal is anesthetized now, so it is unconscious, so we actually move ahead with the practical now. So right now the animal is unconscious, so we actually start with what we want to do. So I'm going to pin the animal to this board. Alright, we have pinned the animal to the board. So this is what we are going to do next. We are going to have a subcutaneous cut. We call it subcutaneous cut because it is going to be a cut directly from the beneath the abdomen, the lower abdomen. So we are going to have a cut in this part. We are going to have a cut in this part. The cut is going to come from this part. So you can actually make do with a scalpel naturally. I should first remove some hair here to make it easy for us to actually do what we want to do. Alright, so let me go in for the opening now. We are actually having a cut from underneath the abdomen now. Let's try a scalpel. So we should try to go as gently as we can in order not to injure any internal organ. Now, for this kind of scissors, it has a blunt edge and a sharp edge. Whenever we're working on something like this, we use the sharp edge outside the body of the animal while the blunt edge goes into the body of the animal. If we insert the sharp edge into the body of the animal, there's a possibility of this sharp edge puncturing into any of the organs. So we use the blunt edge inside to make our cuts. So there are so many internal organs that we don't want rupturing. So we need to be very, very careful. Alright, so we have opened up the animal. Let me pin this. So let us look at the structures that we have here. Here, we have the intestine. This is the large intestine. And here, we have the small intestine. This is the small intestine, as we can see, while this one is the large intestine. So this leads to the rectum. Now, this here, this is the liver of the organism. This is the liver of the organism. So I am going to go, and here we have the kidney. This is the kidney. In a few minutes, we will be dismembering, we will be actually dissecting all of these parts. So there we have, here we have the kidney, and this is, we have a fetus in the uterine lining here. We have a fetus in the uterine lining. We will open that in a short while. So we have here the liver, and close to the liver, we have the gallbladder. The gallbladder, we can see it here. Close to the liver, we have the gallbladder, in which we have what is called the bile. So that is that about this part. So let us quickly, okay. This is a tube that leads from the kidney. It actually houses the urine, so urinary duct. So that is it. And here we have the spleen. We have the spleen here. So that is about that. So let us quickly see what we have in this thoracic region of the animal. So I am going to make an insertion here to have access to the thoracic region of the animal. So we are making an opening here, so we can have access to the thoracic region of the animal, to the thoracic cavity. Alright, so we have so many types of syringes. This is an example of a 2 ml syringe. We have a 1 ml syringe, but what I want to use right now is a 10 ml syringe. So I will quickly use this to get out blood from the heart of the animal. We call it cardiac puncture. Getting blood out of the heart of the animal, we call it cardiac puncture. So that is what I want to do right now. So we are getting the blood from the heart now. If we look closely, we notice that the heart is still beating. Let me turn the blood so we can actually see what I'm talking about. If you look closely, you can see that the heart is still beating. So for the heart to be beating, it means the animal is still alive. So we want to get out the blood from the heart right now. That is what we are after. So we gently insert the syringe into the heart, and we clearly gently stir while we pull out or to draw the blood from the heart. So we gently stir, because if we rush that, we will not be able to get the blood out. So we gently insert the syringe, the needle, and we stir gently. We're just trying to massage the heart while pulling the syringe out gently, so we can get the blood actually from the heart. It is called the cardiac puncture. So that is what we will do until there is no blood left to draw from the heart. So let me invert this so I'll be able to do it properly. From a rabbit on this side, you should be able to draw close to about a syringe, a 10 ml syringe full of blood. So let's see how far we can go from this animal. Alright, currently we have close to 5 ml, but we should still be able to get more. Alright, so this is about the amount of blood we could get from the animal. Let's see. Almost 6 ml of blood. That's the amount of blood you could get from the animal. So if you look at the animal now, you notice that the heart is no longer beating like it was, because we have taken out life from the animal, which is the blood. Taking out blood from the body of an animal. So this signifies that every animal that loses blood, is an amount of blood that every animal can lose. If it loses more than required amount of blood, then the animal dies. So before now, even when we had opened up the animal, the heart was still beating, signifying that the animal was still alive. If we had decided to put back the body of the animal and then we suture the cut part, the animal can survive it. But since we have drawn out the blood from the body of the animal, and then life ceases to exist in this experimental animal. Alright, so we have the animal exposed now. So let me quickly show us some internal structures that we have there. So this one that we have here, this is the lungs. The lung of the animal. It is found in the thoracic region of the animal, so it is with this that the animal respirates. And here we have the heart. So let us try to dismember now. So I want to put out all of these body structures. I want to put them out. Don't want to pierce any of these organs. So we need to be as careful as possible. That is out. Let's go for the liver. trying to get out the liver right now. The liver is very soft. In fact, all the organs, they are soft, so we need to be very, very careful. We have to take them out as a whole. Right here, we have the liver and the kidney. So this is a kidney. So this is a kidney. This is one kidney. Let's search for the other. So here is where we have the gallbladder. You can see the gallbladder here. So it houses the bile. Alright, this is the second kidney. So we have the second kidney now. So these are the two, we have said it several times that the kidney is being shaped. So we can see them being shaped. And here we have the liver with the gallbladder beside it, behind it. And so let us go for the heart now. For the heart, we are going to have to cut open this thoracic cavity. We need to create more room for us to be able to get the heart out in whole. So that's what we are doing now. Right, so we have more room now to work on the heart. So let me use my forceps to pick the heart a bit so I can actually have access to cut it out. Okay, so we are cutting out the heart now. So this is the heart of the animal, and then here we have the lungs. So here we have the lungs of the animal. So these are the kidneys, the liver, the gallbladder, the heart, the lungs. So here we have the intestine, and then the small intestine here and the large intestine. So basically, those are the things that we have there. I want to believe that we are able to get one or two things from all that we discussed here. You understand what dissection is about now and how you can actually get out your organs. So that's how far we can go with this dissection. We're able to start the dissection after anesthetizing our animal and pinning the animal to the board. And then we had to make an incision through the flap, the abdominal flap. So we made a mark under the navel of the animal, under the abdominal flap, and we cut through using a system called the subcutaneous cut. So we cut right under the flap of the abdomen, and that gave us access into the abdomen of the organism. After which we were able to locate the organs. We talked about the organs, the liver, the kidney, the gallbladder, the spleen, and the rest of those. And then we were able to move upwards into the thorax in one piece. We got out the two bean-shaped organs in the body of the mammal, which are called the kidneys, and then we got the liver as well as the gallbladder right beside it. We also were able to get out the heart. This is the heart here. You can see the heart, and then we got out the lungs as well from the thoracic region, and then we went further to get the brain from the cranial box in the head of the animal. Alright, so this is as far as we can come with our practical today, our practical on dissection with the experimental rabbit, which is our experimental animal. That is the Oryctolagus cuniculus. That is the scientific name of the animal. So please, I'll encourage you to try as much as possible to go through the video again to get to understand all that has been said. You need to listen out for keywords of terms that have been used in the course of the practical session. I'll also encourage us to try as much as possible to please subscribe to the channel. If you are yet to do so, subscribe to the channel, as well as you clicking on the notification button so you get easily notified of subsequent uploads when we put them up. So don't forget to like our content, share them, and we want to hear from you. So please drop your questions in the comment sections, and whatever question you want to ask, please put them up in the comment section, and we'll do well to get back to you on them as soon as possible. Thank you very much for always being there, and God bless.